Well, hello everyone, welcome back. As promised, here's our video on the process of meiosis. This will likely be our last video in our Intro to Genetics series, finally. So let's dive right in. So before we get to the actual process, I want to say a couple things real fast about what is our whole purpose for this. Why do our bodies undergo the processes of meiosis? Well, the whole point, as you can see from this diagram here, is again to produce these gametes. So sperm for males, eggs for females. And that is the purpose of this. Now, again, I want to reiterate that these are gametes. These are sex cells, and they are different than your regular somatic body cells in that they are haploid. They only have half the regular number of chromosomes, as opposed to body cells, which are diploid or 2N. So in humans, again, just to reiterate very quickly, your gametes, your haploid cells, they would have 23 chromosomes, and the diploid cells would have 46. So the question is, how do we start out from a diploid cell here and wind up at the point over here where we have haploid? How do we get from here to here? How do we cut these numbers in half? Well, let's dive in and find out. And as you can see here, meiosis is broken up into two big main categories here, meiosis 1 and meiosis 2. So meiosis 1, we're going to start out with a diploid cell. And that is this right here. Now, obviously in males and females, these diploid cells are a little bit different, but we're going to say that this is a male for right now. This is a cell that is going to become, eventually, through this process, 4 haploid cells, haploid gametes that can become mature sperm cells. So we're going to start out this red stuff here I have drawn in to represent the long and thin chromatin, the DNA, the form that it's in prior to this process starting. This would be interphase before we jump into meiosis. So we're going to do this whole thing twice. So this is meiosis 1. So here is prophase 1 of meiosis 1. And you'll notice now that our nice, pretty chromatin has condensed into chromosomes. And you'll notice I have actually color-coded them a little bit. Now, in reality, I probably should have added some more colors in, but we're just going to stick with these 4. It made things simpler for me to draw. They have condensed into their chromosomes, just like it would in mitosis, but you'll notice they are paired up. Instead of just kind of floating around, doing the kind of their own thing individually, they are actually paired up. And there's a reason for that, and these things are called homologous pairs. Homologous pairs. Now, that's very important. That's a very important concept to this. Homologous pairs are chromosomes that match. They go together. Again, the prefix homo means same, so they're, instead of being analogous, they are actually homologous. They contain the same types of genes. They are not identical. They just contain the same types of genes. And so really what we're looking at here is this red one would have potentially, let's say it was inherited from this man's father, and its matching pair, this other shade here, is the one that was inherited from the mother. Same thing here. This one was inherited from father. This one was inherited from his mother. So they are the matching chromosomes that go together. So maybe on these genes right here, that maybe there's, on these chromosomes, there's the genes for height and hair color and eye color, whatever. They're not really together on those genes. I'm just giving an example. So the same things are contained there. Now there's another word that is used for these, a homologous pair. It can also be called a tetrad. Now why tetrad? Where does that word come from? Well, if you look at tetra, that means 4, right? Greek prefix means 4. It's where they get the name Tetris for the video game, because all the pieces in Tetris are made up of 4 blocks. So tetra meaning 4. And if you look at this homologous pair, remember, these could be split right down the middle, and you have each half of a chromosome is a chromatid, and so you have 1, 2, 3, 4 chromatids in one homologous pair total, so it's called a tetrad. Okay, now a very special thing is actually occurring at this point in the process. These homologous pairs, when they get together, they actually touch and overlap right here, and they'll touch each other and kind of overlap in different ways. And what will occur is that they're actually it's a very sophisticated process. There's little enzymes that will cut the DNA at certain sections and actually swap different versions of genes across these chromosomes. So this process is known as crossing over. And what it does is basically we're not mixing up genes that don't go together. We're not taking genes for eye color and mixing them with genes for hair color, but we're basically just shuffling the cards in the deck and saying, okay, well, maybe on this red chromosome over here, there was, the gene for brown eyes, and over here on this other one, the gene for blue eyes, and those swap places so that now the red one contains the gene for blue eyes and the other shade, the one for brown eyes. So it's not mixing up information that doesn't match and correlate and go together, but it is rearranging what versions of the genes that are found on this chromosome are there. So it's like shuffling the cards in a deck. This is super, super important. This contributes to so much genetic variation, which is a big deal. In fact, I'm going to write that word on here. Genetic variation. That is super key. That's like the whole point behind sexual reproduction for many species, is that it increases their genetic variation. When you are reproducing sexually, half your genes, half your chromosomes come from one parent and half from the other, so that you get a lot more variety, a lot more combinations possible, which is good if you're an organism, a species that is out in the wild, and you need to have a lot of diversity so that you could have some organisms, let's say a disease comes through that their genetics make them susceptible to, well, with lots of variation, that there would likely be some in the population that would be immune to it. And so variation is a good thing. And so let's move along. So after prophase 1, just like mitosis, it's going to go to prophase 2, but a little different than mitosis. We're going to see here that in metaphase, I think I misspoke there. Prophase 1 goes to metaphase 1, just like mitosis, it goes from prophase to metaphase. And what we see here, though, is that the homologous pairs are still together. They have lined up at the center of the cell right here as pairs still. And so that's a little bit of a unique feature that's different. But from there, we'll go from metaphase into anaphase, just like mitosis. But again, here it's a little different because we started off not with kind of single file chromosomes, but buddied up, paired up chromosomes. And so those homologous pairs are going to separate in anaphase. They'll jump right into telophase 1. In telophase 1, we're going to start to get this pinching in here, this cleavage furrow that's forming. And shortly after this, cytokinesis will begin. But we see here the nuclear membrane is kind of coming back. These actually, I didn't draw it this way, but these would probably be uncoiling back into chromatin. It's just normal telophase stuff. And it's followed by cytokinesis. So what we have at this point, after this right here, after meiosis 1, we end up with two cells that are both haploid. They are both already haploid because they have half the regular number of chromosomes. So over here we started, there were 4 chromosomes. All right. Now again, they're copied, but look right here. This is already cut in half. Okay, we've lost the number here. We've cut it in half, but that's on purpose because our end result is going to be 4 haploid cells, okay? So after cytokinesis, it's going to jump right into meiosis 2, and it's going to follow the same steps, the same phases in the same order. So we go into prophase 2, but again, both these cells are doing this side by side. There's no crossing over or anything like that that's occurring now. This is just plain old, normal, dividing up the nuclear material like mitosis. It's just meiosis 2. And these chromosomes are not identical because of crossing over. So we go to metaphase 2. Now they're lined up single file. Looks like mitosis. Continues on into anaphase 2 right here. Notice what's being pulled apart here? Now we've got sister chromatids that are separating. Okay, so this is like normal. We go on, telophase 2, we get that pinching in, that cleavage furrow, they're starting to form cells, and by the time cytokinesis is done, here is our end result. Now, when you look at the way I drew it, I really probably should have used a little more color, but it would have been rather tedious to do. What it appears at first glance that, you know, this cell and this cell, it appears that they would have the same information, but they do not, in fact. So what would have happened up here when, you know, this red chromosome and this pink chromosome back in prophase 1, when crossing over was occurring, they exchanged portions. So really, I should have drawn, you know, like some of this shade tucked over here on this chromosome, and then some of this red tucked over here on that chromosome to show that they had crossed over. They just would have been too tedious. So what we actually end up with is that these cells down here, all 4 of them, they're all 4 unique. They are not identical really in DNA in any way. So 4 unique, again, haploid cells. All right. Now, we said this was a male, and so in this male, all 4 of those will develop, if everything goes according to plan, all of those will develop into mature sperm cells. However, what if we had said this was a female? A little bit different here. It would have actually occurred if this were a female that only one, take your pick, any of the 4, one of the 4 would have been lucky. Only 1 out of the 4 would have become a mature egg. The other 3 literally would have just died off. And those other 3 are what are known as polar bodies. Again, that's in a female human. Only 1 of the 4 cells that is produced becomes a mature egg that could be fertilized and form a new human. All right. And so real quick, before we head out, I want to just recap some of these major terms. Homologous pairs and tetrads. These are matching chromosomes. One comes from mom in each pair. The other comes from dad in each pair. They are homologous. Homo meaning same. So they have the same types of genes, these same types of informations. They're also known as tetrads because there are 4 sister chromatids in each pair total. When they get together, when these homologous pairs get together, it leads to this event here, crossing over, which again we said is huge. This is what leads to genetic variation. All right, lots and lots of variation. It's an insane, like astronomically high number. I don't even remember how large it is. It's just ridiculously insane that if 2 humans could somehow, you know, combine their sperm and eggs. I mean, it is just, it's insane the number of zeros that are on the number of how many times they could just like naturally conceive and never get 2 identical children. It's ridiculous. So lots and lots of variation there. And it occurs twice. So we're going to go through our process of nuclear division where we divide up the nuclear material in the cell. Again, PMAT, prophase, metaphase, anaphase, telophase, but it's different than mitosis because we're going to do that twice. That's what gets us for a total of 4 cells, which again is our end result. The whole purpose, the whole shebang here, this is what it's all about, the end result. 4 haploid gametes that have different DNA. All right, well, guys, I hope this video has been helpful to you in trying to understand the process of meiosis. As always, feel free to like this video, comment below, subscribe to my channel. Let me know kind of what videos you guys would like to see that you would find helpful to you in your high school biology class. Thanks for watching.